is stereotypically a lot more difficult for students. And so at this point, if you haven't mastered that study technique or how you should be approaching these chapters, this is really one that you need to focus on. There's just far too much information. And for most people, it's not that intuitive. I mean, photosynthesis, if you think about what you may have learned before, we talk about, how we take sunlight and make sugar. Now, obviously, it's not that simple. We don't get as complex as it truly is, but there needs to be a basic understanding of it. So there's a certain level of detail when it comes to the reactants and the products and how the chemical reaction is occurring that we do need to understand. And so what I'm trying to say in a lot of words here is this is a chapter that if you haven't already, you need to start focusing on at least 10 concepts a night or so. Look at a couple slides every night or day. I don't care when you do it. You're in charge of your own life, but you need to be looking at some of these to make sure that you don't get behind. This is not a chapter that you can simply wait until the night before and cram it all in. I have broken up the units a little bit differently than we previously did. And so this semester, chapter 7 and 8, photosynthesis and cellular respiration, are in their own exam. So a lot of students liked it last semester because it allowed them to focus on these two difficult chapters, but that does mean that everything that I ask on the test will come from these two chapters. And so anything that's in the PowerPoint or anything that I discuss is going to be important. So let's just start with what the basics is. What do we think photosynthesis is? Or what have we heard before? Well, in some way, there are life forms that can take solar energy and convert that into a potential energy source, into a sugar. Now, we're not taking sunlight, which is just waves of visible and invisible radiation, and converting that directly into a sugar. That's impossible. But what we can do is transfer the energy from one form to another. So what we can do is well, not we, but what plants and other photosynthetic organisms can do is take solar energy, take this radiative energy, and charge up some electrons, and then store those high energy electrons. And then when they need them, break that down and access those electrons. So that's what we learned last chapter. That's all glucose is a way for us to store all of that high energy in that compound. And so we say plants make sugar from sunlight. That's the basics. What we're doing is we're transforming that energy into chemical energy of carbohydrates through photosynthesis. And now photosynthesis doesn't just end up in a carbohydrate, doesn't just end up in a sugar. I mean, we can make amino acids, we can make lipids, all kinds of other metabolites, but we say photosynthesis ends up with carbohydrates, and so we transform that into a chemical energy source. If an organism is capable of manufacturing its own energy source, its own sugar, its own food, through solar energy without consuming something else, we call it an autotroph. Auto means self, troph is referring to that energy level, trophic level. And so they're producing food and energy through photosynthesis. So we're talking about our algae, plants, cyanobacteria, usually your green things. Not all green things. There are a couple different types of photosynthesis that has evolved over the millennia. But usually your green things are your photosynthesizers, they're your autotrophs. If something has to consume another organism in order to access some of these sugars, in order to access this energy or amino acids, we call it a heterotroph. Hetero meaning different, troph referring to that energy level. So heterotroph must consume or acquire energy. And so autotrophs, using that solar energy, can make sugars, make food, make an energy source, amongst other things, that heterotrophs have to consume. The process of photosynthesis also has a byproduct of oxygen. It's not its main purpose, it's just the molecule that is released during the photosynthetic chemical reaction. And so we benefit from that. Photosynthesizers are what have transformed our atmosphere into the oxygen-rich environment that we enjoy, right? Nitrogen won't work in our lung system. We have to have O2. And we'll talk about why that is later down the road. But Heterotrophs, here's a horse. Why did they choose a horse? I don't know, but it's a heterotroph, right? Consuming photosynthesizers. And then we'll talk about cell respiration next chapter, which is where we take the stored energy, access it, and a byproduct of cell respiration is carbon dioxide and water. And so it's a cycle. These are two equal, well, not necessarily equal, but two partner reactions, photosynthesis and cellular respiration, as far as the balance that we are used to. And so now we got to get into the nitty gritty. If there are some definitions that you didn't learn last chapter, now is when you really have to know more. So photosynthesis is taking place not everywhere in the plant, but if we can zoom in at that molecular level, it's taking place in the green portions. So what makes it green? Hopefully this is review, but the cells that contain chloroplasts, and we learned that plast is just means membrane bound, right? Plastid. And so if we're zooming in on a chloroplast, there are specialized areas called thylakoids that carry out photosynthesis. Thylakoids are packed full of a green pigment, a green protein called chlorophyll, which just means green leaf. Chlorophyll, chloros, green, fill, leaf. And so these are proteins that are capable of capturing solar energy, speeding up some electrons, and then storing them. Where else do we hear thylakoid? Well, in the last chapter, we learned that there are certain cyanobacteria, certain bacteria that are wrapped in a thylakoid that can perform photosynthesis. And so inside of a chloroplast, what you essentially have are these factories of very, very, very primitive, degraded bacteria. You can call them endosymbionts. I mean, they're no longer bacteria. They can't live on their own. They've been highly modified. My mouse keeps disappearing. Highly modified into these stacks that we call granum. But thylakoid specifically is where the chlorophyll is located, where that photosynthesis can occur. It's thylakoid, not thycoloid, not flacoid. It's thylakoid. And remember, I said some of you kind of just go, I can't pronounce that. Blah, blah, blah. You know, you look over it. That's hurting you study-wise. And so sometimes I have you say things out loud. Since we're not in class, I can't have you do it, but I'm imagining you're at home right now. So if you are, or wherever you are, just say the word thylakoid. Once you understand how to pronounce it, your brain won't skip over it when you read it. And so it kind of loses that intimidation factor. You stop saying like, this is just too much, too confusing. I can't get this. These words are too big. Well, yes, you can, right? You've learned words before. You can learn another one. Thylakoid is just that membrane where photosynthesis occurs because of the chlorophyll pigment. In order to understand photosynthesis, you have to start with the basics and then build yourself out. Start with the basics of this reaction and then learn the heavy details. So the basics is a photosynthetic organism requires carbon dioxide. That's where your carbon's coming from. Look at that formula, CO2, and water, H2O. So in order for photosynthesis to occur, an organism has to have access to carbon dioxide and water. We focus a lot on plants in this class. We don't focus a lot on bacteria or algae or other photosynthetic organisms. Sorry if you heard that. I don't know if that was a truck or thunder or why that's odd. But carbon dioxide and water. Well, where does water come from? They absorb it through their roots, through through the other cells along the leaf. But where does carbon dioxide come from? Well, think for a second. Carbon dioxide is a gas. And so that gas is all around us right now. So how is it going to get into the cell? Well, it can diffuse in. We learned that gas can diffuse right through a membrane, right? That was the whole point of last unit. And so the gas can move from an area of high concentration, which is all around us, to low concentration. There are special locations on the leaf that this diffusion can occur in high amounts, openings that we call stomata. A stoma means an opening. So a stomata is a singular opening, stoma plural. It diffuses into chloroplast, the CO2 does, in special middle cells of the leaf. Now, inside of the cell, we said that fluid-filled substance was the cytoplasm. Inside of the nucleus, we call it the nucleoplasm. Inside of the chloroplast, the liquid fluid substance filled with enzymes, DNA, ribosomes, starch granules, and those thylakoids is called the stroma. So a lot of words that sound a lot alike here. Stoma, stomata, stroma. So you'll probably need to write these down a couple times just to make sure you don't get confused, because that's the difference between an R and no R, right? Very similar. But stroma is the fluid inside the chloroplasts. A chemical reaction requires energy. These things don't just happen. These molecules have to, in some way, have enough energy to collide and react. Where does that come from? Well, the energy is supplied by light, by sunlight. Not necessarily sunlight, it could work from a lamp source if it's the right amount of energy. Chlorophyll and other pigments absorb solar energy and energize electrons prior to the reduction of carbon dioxide to a carbohydrate. Well, at the end of last chapter, we learned that reduction means gain of an electron. And so what we're doing here is we're taking carbon dioxide and water, using an energy source supplied usually by sunlight, and then taking that to energize electrons. Well, when we energize them, we don't want to waste all of that, so we store them somewhere. So something has to store those high energy electrons, and so we end up storing into these carbon compounds, and that makes our carbohydrates. So this is all occurring in the green portion of the chloroplast, where the thylakoids are. And so CO2 can move in, providing the raw material we need for carbon, as well as providing O2, which allows us to store our high energy electrons after we've used them. You'll need to memorize the formula for photosynthesis. And so the formula is we take carbon dioxide and water, and we end up with glucose and oxygen. When you have a chemical reaction, we have to follow our rules of physics here. Whatever we have on the left side, what we start with, we have to end up on the right side. Things can't just disappear. That doesn't make sense. Those molecules have to go somewhere. And so a chemical reaction allows us to write down how many we have. And so for photosynthesis, this reaction is 6 CO2 plus 6 H2O gives us C6H12O6 and 6O2. And so we started with carbon dioxide, water, ended up with glucose and oxygen. This requires energy. These two molecules aren't just going to be able to do this process, right? There are proteins and substances and enzymes that are at work here. So they require energy, they require ATP. So where can we get that energy source? Well, in this case, to start out with, the energy source is the sun, or solar energy, or high energy visible radiative energy. It's what we call an oxidation reduction reaction. If you read the last part of the previous unit, you'll know that oxidation reduction means something is losing an electron and something is gaining an electron. So let's look at it. So if we take CO2 and we have to move those around and manipulate them, we're going to be removing some electrons. Where are they going to end up? Well, we store them in glucose. That's the whole point, is to create, and store a high energy compound so that the plant can use it when it needs an energy source. And we just benefit from that because we consume it. And even if you never eat plants, you've eaten something that's eaten a plant. Or if all you eat is Cheetos, you've eaten a byproduct of something that used to be a plant, right? And so in this chapter, it's a little heavy on some chemistry. And so there are some basics that you have to know. You have to know that when you see the formula C6H12O6, that's glucose. You should know that backwards and forwards. So whether I'm saying glucose and you have to come up with the formula, or I give you the formula and you have to know that it's glucose, know both ways. That holds true for oxygen, water, and carbon dioxide. When you're reading questions or when I'm discussing things, you should not have to think about what these two mean. It should just be second nature, CO2, carbon dioxide, H2O, water. Now the rest of these, there are a couple that we really focus on and a couple that we don't. Please don't panic with these long organic molecule names. Some people need to know that in the future. Some of you will never need to know it ever again for the rest of your lives. And so I put both, but notice I say we only need to know abbreviations. And so we start off with NADP, which is going to allow us to carry some electrons. We'll make a special molecule called G3P. RUBP is a special sugar. And so we'll become familiar with these. But the first four, know name and formula. These, just understand that these are molecules that we're going to be understanding. And if you want to know the names, here they are. If you're taking notes, you may want to make a little note at the end of this one. It's too late for me to type it here, but we've changed the rules of how we name some organic molecules. We've made it a little bit easier to understand the chemical structure from the name. Glyceraldehyde 3-phosphate used to be called phosphoglyceraldehyde. Why do you care? You don't. But sometimes we'll do assignments that instead of saying G3P, it will say PGAL, PGAL, and I'll point that out. But phosphoglyceraldehyde and glyceraldehyde 3-phosphate, it's the same thing. The 3 just lets us know particularly where the phosphate is branched from. So it helps us draw the structure if any of you ever have to do that. That was thunder. I didn't even realize it was storming. Let's go to the Weather Channel app, right? Is anybody still on here? Let's see. Yeah, we got 4 participants. So let's see. Thunder and ice, that's a weird combination, right? Doesn't even say thunder. It says ice and then wintry mix. Who knew? Think we'll have school tomorrow? I don't know. We'll see, right? 97% chance of precipitation. So, maybe we don't go anywhere today unless you really have to. You know what I'm going to do? I'm going to post this, right, on YouTube, and then I won't update it for the next 3 or 4 years. People will be listening to me like, oh, they had thunder back in 2022. Anyway, let's move on. I do get distracted. So we have to cover 2 main processes of photosynthesis, the light reactions and the Calvin cycle. And odds are we won't get to both of them today. This chapter takes a very long time, often three lectures. For the Tuesday-Thursday class, it'll probably take close to two and a half. So we focus on the light reactions first. And so I like to separate that in my head. In fact, when I studied this chapter, I had a white piece of paper or a notebook paper, and I would draw out the 2 reactions. The first thing we do is the light reactions. It requires light. So sometimes we call it light dependent. And then on the right-hand side of my paper, I would draw the Calvin cycle. Sometimes we call the Calvin cycle the light independent because it does not require light. We make all the energy that we need on the left-hand side. The right-hand side is where we end up with the products and use the energy we made. The Calvin cycle reactions used to be called the dark reactions. We thought that if one occurred in the light, the other occurred in the dark. We know that's not true. Melvin Calvin, if you're reading your textbook, discovered the Calvin cycle reaction by synthesizing some of the products that we end up with. And so he was a brilliant botanist, and chemist, able to understand that just by what we put into a reaction and what we get out of a reaction, he mapped out what the Calvin cycle was. We couldn't actually see it taking place, and we still can't really see it take place. We can zoom in and get close and watch some metabolites, but we don't really see it. We just know what we end up with and what we start with. And so because he was so brilliant and really helped us understand these reactions, we honor him by calling it the Calvin cycle. But it's really the light independent portion of photosynthesis. The light reaction, we'll talk about where the energy from sunlight goes, how chlorophyll absorbs it, how we charge up those electrons, how we make some energy compounds. And the Calvin cycle is essentially where we make the sugar, or we make the carbohydrate, or other products, if you ever go down that road. So how do we know photosynthesis occurs in a plant? Well, we could take some of that LOD we looked at in lab, we could put it under a lamp and put that under water, and eventually we would see bubbles forming all along the leaf. Well, what's happening? It's taking in carbon dioxide, taking in solar energy, making sugar, and producing oxygen that diffuses out. That oxygen, right, is a gas And so it has a higher volatility in the water around it. And so it forms this bubble inside of those water molecules. And eventually, right, as the pressure inside that bubble gets higher, it's going to go to the surface and, diffuse on out or mix with the water as well. And so oxygen gas is formed at the end of photosynthesis as well. And so where are we occurring? If you're writing out your practice sheet that you have under resources, or you're writing it on a notebook paper, the light reactions are occurring in the thylakoid. Do not forget that. The green portions, that's what the chlorophyll is. That's where the light reaction is occurring. The Calvin cycle is occurring in the stroma, out in the liquid portion, because that's where we're making the sugar, where the enzymes are. We don't have to absorb sunlight. Things are green because we're absorbing that sunlight. In the Calvin cycle, we're just making the sugar. We're using the product of the light reaction. That's what powers it. So essentially, the light reaction makes batteries that power the Calvin cycle that makes the sugar. So that's how we can go from sunlight to ATP, NADP, H, and then form glucose or amino acids or whatever. CH2, that's just kind of like a general organic molecule. So let's discuss how that happens. Pigments are proteins that are made up of molecules that absorb certain wavelengths of light. What a wavelength is, if you can picture a wave, are a little, well, a photon is a packet of energy that acts like a wave that our eyes can pick up. If we're seeing a color, that's the wavelength that's being reflected back to us. So every other color is absorbed. So look at what shirt you're wearing or what desk you have or whatever it might be. That color you see is being reflected. What is being absorbed is everything else. And so there's a certain absorption spectrum. Certain pigments are able to absorb different wavelengths. Now, there are also wavelengths of light that aren't visible to us. So microwaves, radio waves, X-rays, gamma waves, things that we can't see because the cones and rod cells in our eyes don't pick up on those things. And so we can only see visible light. That's all we needed to see to keep us alive. We don't need to see the others. So chlorophyll is green because it's absorbing the reds and the blues, and the green is reflected. Well, that seems like a waste. Why would you not just absorb all? Why would the chloroplast not just be white? Because it does the job, right? It all comes down to evolution, and it works well enough, right? It's great, it's fine, it does what it's supposed to do. But it seems like that's a waste of excess light. So if a plant or an algae or a cell, whatever, that's photosynthesizing does require more energy, some have accessory pigments, things that help out the chlorophyll. So as an example, one that we say a lot are carotenoids. Well, what does carotenoid make you think of? Carrot. Carrots are oranges. So carotenoids are reflecting some of that yellow-orange light close to that green spectrum and absorbing violet, blue, and green. And so there are other pigments beside chlorophyll. It's just that chlorophyll is in such high abundance and is able to absorb that energy so well that it does fine without that green light. And so there are accessories, though, that can help pick up on that. And so the important accessories that we have to know, the important pigments, are chlorophyll A and chlorophyll B. They're going to absorb violet, blue, and red light and reflect green. And then carotenoids. It's a very important accessory pigment that absorbs violets, blues, and greens and reflects oranges. And so it's passing energized electrons to chlorophyll A. This mass of colorful pigment absorbing solar energy is called a photosystem. It's like an antenna that can pick up energy waves. And so if you've never had physics before, or this is new to you, use these pictures to help you out. Here's the light spectrum. Picture light as a little packet of energy. That's very basic, but imagine a little tiny microscopic, impossible, weightless packet of energy that exists as a wave. So it's waving through the universe as it moves through. The small, really short waves, gamma rays, X-rays, UV, we don't see that. We see the small spectrum here, visible white or visible light wavelength. Now there are longer waves too, infrared. We can we utilize to see heat, microwaves and radio waves that we utilize for communication. But we can only see these, and so plants, yeah, they're absorbing some UV. They don't really absorb infrared, but they're absorbing some UV, but a lot of visible light. And so this chart here puts the visible light spectrum down here. It goes from 380 to 750 nanometers. That's the size of the wavelength, and shows us what is absorbing what. So your chlorophyll A, the dark one here, is absorbing a lot of violets and blues, and your oranges and reds. So if you go to the store, we're all really into plants right now, the house plants. If you get a plant bulb, it's usually bluish or reddish. Well, why is that? Because the light that we use to see, like so the light that I have lighting up this room here, that's more of a bright daylight. That's a lot of white, bright light, a lot of wavelengths. But if you're just trying to get your plant to grow, you don't want to waste energy on this whole spectrum, because look at all of this light that's not being absorbed by chlorophyll. So why would you waste that? But if you focus on the red range here at the end of 600, 700, or here at this wave with the blue, that's a ton of energy that's absorbed by that plant. And so, they make the bulbs in blues and reds to help out. And then there's some accessory pigments, carotenoids, and others, xanthophylls, anthocyanins. There are other pigments, some fat soluble, some water soluble, but we focus here chlorophyll A, chlorophyll B, and carotenoids. When we access solar energy, when it is absorbed by those pigments, and we charge up that electron, well, great, now what? We can use that energy to make ATP, adenosine triphosphate, that high energy, unstable compound that we said can easily break off that third phosphorus molecule and pass an electron along, or use that burst of energy to power something. And so ATP is what we can use to reduce carbon dioxide to a carbohydrate. So we can give it an electron to form that high energy carbohydrate. Now, electrons are tiny, small, somewhere in the range of 2,000 times smaller than a proton or a neutron. We don't want to lose them. We don't want them to lose too much energy. We don't want it to be inefficient. And so we have to have a molecule that can store an electron for a while. And so we use NADP, which is a high energy B vitamin battery. It's technically a cofactor of the redox reaction. So without the cofactor, the metabolic reaction can't continue. And so we have to have the cofactor. But think of it as a B vitamin battery. It's reduced, so we stick an electron to it. It carries that hydrogen, and it can hold that energy for a while. And we need it to power something. We break it apart, remove that electron, remove that hydrogen, and now we have energy to power something and to reduce something else. The light reaction, in order for us to break apart water, get some electrons, and make ATP, there are two pathways electrons can go down. There's a non-cyclic and a cyclic. One that just is, we have to keep constantly resupplying, and one that kind of has its own cycle of resupply. So we're going to capture light with those pigment complexes, those photosystems. Now here's where it gets a tiny bit confusing. We named them photosystem 1 and 2. Technically, we named them PS700 and PS680, which just tells us how much or what the range of light wavelength is being absorbed and what's going on there. But we named them photosystem 1 and 2. Photosystem 2 actually occurs first in this light pathway, and photosystem 1 occurs second. But we discovered photosystem 2 first and photosystem 1 second. And so we just haven't changed the name. Maybe one day, you'll go on and you'll make this less confusing and you'll update the name. But PS2 and PS1 are these photosystems that absorb solar energy and pass those electrons on. And so photosystems capture light energy, they charge up an electron. The electron travels down what we call an electron transport chain. So it's going to go from PS1 or from PS2 to PS1. You can't let an electron leave. And so it's passed from protein to protein to protein to protein. As it travels down this electron transport chain, we can use that energy to do stuff. And so this is the pathway that's allowing us not just to pass a high energy electron to an eventual, to an eventual molecule that we can store it, but it also allows us to use that electrical energy and use that heat energy to do other reactions. Now, and I want to show you all this again with pictures. We'll do it 2 or 3 times, but we access that first electron from water. And so we split apart water, H2O, so we have some hydrogen and oxygens to get an electron, so that we can replace the electron that we're passing down the electron transport chain. And we're going to keep using that hydrogen. And so we're going to store that for a minute. I'll have a picture, so let me get through these, and I'll show you it all again through pictures. So the electron is transferred permanently to a molecule of NADP. We have to store it. The electrons are negative, so we have a bunch of this H plus floating around as a gradient. We stick the H plus and the electron to NADPH to form NADPH, which is that B vitamin battery. So this is like the metal casing of your battery. This is a battery full of high energy electrons that you can use. So your battery dies in something, your battery dies in your remote, you have to replace it. This is the high energy compound, because we can break apart this hydrogen, that gives us a negative electron, and we can use that to power something. So where are we? Remind ourselves. Light reaction is occurring in the thylakoid space. Solar energy, those ions are striking those pigments. Electrons are, I meant to say ionic radiation, but the solar energy is striking that pigment and charging those electrons up. This photosystem, if you can imagine it like a glob of green pigment, when those electrons get heated up, that powers up, those jump up in high energy level. It becomes so highly energized that it leaves. And so it travels down this electron transport chain and it hits photosystem 1, which is like those old physics. Have you seen those physics models with the silver metal spheres that allow you to study energy of motion and inertia? You can pull one back and it hits them, and then the energy travels along all those, and the one on the end bounces back and it goes back and forth, back and forth. That's kind of what's happening here. This high energy electron is getting so charged up from that electrical solar energy that it breaks away. It can't be held on by the positive charge of those protons anymore. It breaks away, passes down the electron transport chain, hits photosystem 1, which is also absorbing solar energy, which then powers up one of those that breaks away. So let's pause on that for a second and go back over here. We can't just keep losing electrons. So in this non-cyclic reaction, where we lose an electron, we have to replace that electron from water. So circle that, highlight that. That's a common question, always in an exam, a common question, always in homework, a common question, always if you're in my online class, common question in a smart book. Where does photosystem 2 replace the lost electron? Well, water. How do we know that? Oh, I can't memorize this. This is too much information. This is just way too much. Well, slow down. Go back to the note that you took that says, what are the raw ingredients of photosynthesis? CO2 plus H2O yields glucose and oxygen. So when we ask a question, where is the electron replaced from? Well, we can't just make something up. All we have available in our reaction is CO2 or H2O. Well, I told you CO2, that's the carbon we're going to use for the sugar. So that's not it. What else do we have left? Where can the electron come from? The only molecule we have left in that reaction is the water. So that's the only thing that it could come from, the only thing we have in this reaction. If it was something else, that would be written out in the chemical reaction. But we didn't. All we wrote out was CO2, H2O yields C6H12O6 and O2. And so water is split apart, H2O. Technically, to get one, you would get one molecule of oxygen, and so they write with the half coefficient in front of it. But O2, because it's a gas, doesn't exist as just O. Otherwise, it would be O negative, and it's not. It's O2 and 2 H2O per water molecule. That electron is replaced. And so then as it transfers down the electron transport chain, this photosystem 1, when its electron leaves, that's a very powered up electron. We're going to store that. in NADPH. So here we have a protein that we're going to call NADPH reductase that's going to reduce NADP to make NADPH. Where is this electron replaced from then if we keep losing it? Well, it's constantly being replenished from photosystem 2 as long as there's light energy available. Now, as a byproduct, we do get some hydrogen ions left over inside the thylakoid. So that's going to allow us to use to make something else. And so photosystem 2 consists of a pigment complex and electron acceptors. When those electrons are powered up and leave, they receive electrons from the splitting of water. O2 gas is released. The electron, we can't just put it nowhere. It has to go down the electron transport chain. So we're carrying an electron between photosystem 2 and photosystem 1. We're going to store those excess hydrogen into the stroma in the thylakoid space. Photosystem 1 also is a pigment complex, also is going to receive those electrons, also is receiving solar energy. But rather than passing its high energy electron down another ETC, we're going to use that to store in that B vitamin battery to make NADPH. Now let's go back to all that hydrogen from breaking apart water. Where does that hydrogen go? Well, ATP synthase is a specialized molecule, a specialized protein that's there inside the chloroplast, inside that thylakoid membrane. That specific function is to synthesize ATP. It has a channel, so I don't know if I can draw anything here. Let's see. Let's use green. So if you could picture that we're here, all along the chloroplast. Here's photosystem 1, here's photosystem 2. And we're passing the electrons along it and passing them out to go. Electron. When we break apart water, we are building up, and imagine I wrote this over and over and over again. We're building up hydrogen ions inside the chloroplast. Well, lucky for us, there's a very special enzyme, and it's not over here, it's really down over here, whose job is specifically to make ATP. Well, how does it do it? It has a channel through it, looks like a little windmill. Water wheel, what am I trying to say here? I was trying to say water wheel and windmill at the same time, and windmill came out. But it looks like a water wheel, and it can specifically fit hydrogen ions. It hits that turbine, and this spins. This electrical energy allows us to take, or kinetic energy, allows us to take adenosine diphosphate and phosphorylate, add a phosphorus and make ATP, adenosine triphosphate. So we can successfully synthesize ATP. I am always bewildered every single semester when a question asks, which of the following enzymes synthesizes ATP? And if I gave you NADP reductase, lactase, amylase, ATP synthase, of those 4, which 4 do you think synthesize ATP? Think about that for a second. Well, probably the one called ATP synthase, right? It's synthesizing ATP. That is the answer. It's not a trick question. I say it like this every single lecture, every single year. I have it written out. People still always miss that question. ATP synthase is the enzyme responsible for synthesizing a majority of ATP, not just in photosynthesis, but also in cellular respiration. So please don't forget that. And so I love this picture in the textbook. I studied from a very similar picture. It lets me see that the thylakoid is just wrapped in this phospholipid bilayer, that we have these photosystem pigment complexes absorbing sunlight, that passing our electrons down the electron transport chain, end up at NADP reductase to reduce NADP, and then we're building up hydrogen ions that can flow across ATP synthase to make ATP. This is the perfect picture to study the light reaction. It's all right here. And so it brings to light what the reaction is really about here. And so what's the purpose? So ask yourself, what is the purpose of the light reaction? Well, if we're going to make sugar, that requires energy. So what we have just done in the light reaction is take sunlight, essentially, charged up some electrons, and now we have NADPH, a B vitamin battery, and ATP, a high energy compound. And so we've been able to take sunlight and make these two high energy compounds that can then charge or power up a cycle that's going to make a sugar. So where are we? The thylakoid membrane. What are we doing? Making ATP, making NADPH. So I highly encourage that you look at that study worksheet and that you fill this out using this picture and using this portion. If you remember from the last part of the presentation, some of you do, some of you don't, chemiosmosis is the word to describe an electrochemical gradient, the movement of electrons and ions down a gradient to power something. So chemiosmosis is occurring in this thylakoid space because we're charging up hydrogen ions. We're filling it like filling a balloon. And that means there are more hydrogen ions on the inside of the thylakoid than on the outside. So we have a concentration gradient. We have a difference in pressure. And we just learned that things are going to flow from an area of high concentration to low concentration. So those hydrogen ions want to leave that space. But they're charged ions, so they're not going to fit through that phospholipid bilayer very well. Where can they leave? Well, there's a pump channel directly through ATP synthase where hydrogen can fit. This is chemiosmosis, building up a gradient to use that and utilize that electrical energy to make ATP, to make energy. So the flow of that back across the thylakoid membrane energizes ATP synthase and allows us to phosphorylate ADP to make ATP, that high energy compound. So where were we? Thylakoid. What did we do? Make ATP and make NADPH. We're not done. We have to make glucose. We have to store that in glucose. So the next part of the photosynthesis is the Calvin cycle, or it used to be called the dark reactions or light independent. So here we have to take those high energy compounds and store it in a big carbon molecule. Where does our carbon come from? Go back to your reaction. CO2 plus H2O. But we already used H2O back in the light reaction. So all we have left to us now are the gaseous carbon dioxide molecules. That's our carbon. That's where the C comes from. And C6H12O6 is carbon dioxide, or at least a portion of it. We're going to really reduce this, really store the electrons, and then we have to kind of replenish some of our ingredients. We have to go back to the store and buy more ingredients, essentially. And so it involves 3 stages. We have to take the gas and make it not a gas. We have to stick the gas to something. We have to reduce it, which means gaining a lot of electrons. And then because this requires us to use a special compound called RUBP, we have to remake RUBP. And then we're done. That's photosynthesis. So where are we? We're in the stroma, the fluid-filled portion. This Calvin cycle is a system of reactions where we take a carbon dioxide and we reduce. We've taken that carbon molecule and we add an electron, add an electron, add an electron, add an electron in this cycle. Every time you add an electron, you're bonding other molecules and you're changing the shape. When it changes the shape, we give it a different name. So eventually we go through this process where we bind CO2 to a molecule and it changes until we get this nice, perfect carbon skeleton called G3P. So let's see where we're at. So the first step of Calvin cycle, of the Calvin cycle, is fixing that gaseous carbon dioxide to a solid source, sticking it, gluing it to a solid source. So we take CO2 and we attach it to a 5-carbon sugar called RUBP, ribulose biphosphate. We always just say RUBP in this class. Things don't just happen. It would take a lot of energy for CO2 to stick to RUBP in the right shape and the right orientation in order for it to fit. What do we do? What can we use to help lower the energy of activation and help us stick things together? Use an enzyme. We just learned that. Time to go back and refresh before you start this one. but we use an enzyme to do that. The enzyme that allows us to stick CO2 to RUBP, we call it Rubisco. Kind of sounds like Crisco, right? It's kind of a fun name. Technically, it's called an RUBP carboxylase, but we say Rubisco because that's easier. So Rubisco is the enzyme that helps us stick CO2 to RUBP. The CO2 or carbon dioxide is now fixed because it's part of that. It's a solid source now. It's now a 3-carbon molecule that we can attach some electrons to. What's it called when we attach an electron? It's called reducing. So we can now reduce that carbon. So that 3-carbon molecule is reduced to something we call BPG from the NADPH that we just created. And we can reduce it again from ATP and make glyceraldehyde 3-phosphate, or G3P. This is a carbon skeleton that can store energy, and we can form big molecules like glucose. We can make amino acids, we can make lipids, we can make fructose. There's a lot we can make from G3P. Glyceraldehyde 3-phosphate, big, large carbon organic molecule that we can use to make a bunch of other stuff. It's kind of like, I always think of photosynthesis like a recipe. G3P is kind of like flour, right? Nobody really eats cooked flour, but it's a universal ingredient that we can use to make cakes, cookies, gravy. You can use it to thicken things, and you can use flour for all muffins, right? All kinds of things you can make from this. So G3P, we can make all kinds of things out of it, not just glucose. So really the major end product, the big thing we want to make at the end of photosynthesis is truly G3P, glyceraldehyde 3-phosphate, or for those of you that need to pause this, it used to be called phosphoglyceraldehyde, PGAL. We call it G3P. It utilizes some NADPH because that's where the electron is going to come from. Some ATP is produced in the light reaction, so we use some of those electrons. And so we make some G3P, but unfortunately, this molecule that we use to stick CO2 or to fix CO2 is expensive. So most of the G3P that we make in carbon reduction can't go on to make glucose. We have to take it and use it to remake G3P or remake RUBP. And so this picture I like. It helps you fill out the study worksheet. You can take ATP and it shows you that we reduce, there's the electron, we reduce, there's the electron, to make G3P. So the third and final step of the Calvin cycle is to remake that RUBP. We have to regenerate it, otherwise we'd run out and we couldn't keep doing photosynthesis. And so RUBP that is used in CO2 fixation has to be replaced. So for every 3 turns of the Calvin cycle, really you need 5 of your 6 G3P to make 3 RUBP. So it's expensive. Photosynthesis is very expensive and extremely inefficient. However, it is more efficient than anything we have ever been able to create ourselves to make energy. But it's wildly inefficient and can build up toxic byproducts. It's not a perfect situation, but obviously it works well because this is the main source of energy formation on the planet. And so when we get to the natural selection and ecology chapter, we'll learn that some things that work well enough are what's going to stick around. Well enough is a very, very, very common theme in biology. It works well enough. Like our lungs, right? Weak. If they get clogged, they collapse, we die. We go into water, we die. They can only use oxygen. They can't use carbon dioxide. They can't use any of the noble gases. They can't use any nitrogen, which is the major gas component of our atmosphere around us. They're weak, but they work well enough. And that's pretty well, right? They do a pretty good job for what they do. So photosynthesis is like that. It works well enough. And so even though we have to remake this RUBP, even though we have to take G3P to remake it, it works pretty well. So at the end, we get about 5 G3P in ATP and 6 NADPH from light reaction, carbon fixation, and carbon reduction. But we have to use those just to remake RUBP. So here's the point where if you're filling out that study worksheet, you should probably stop. Don't stop listening to me, but when you go back and relook at this, take that paper I told you where you had the light reaction and Calvin cycle. Hopefully you have written down or will write down where they occur, what the product is, why. And so here's where you can think about the why. You have to understand the why to help you. You're going to learn that biology isn't just memorizing vocab words. It kind of gets that reputation in middle school, sometimes in high school, but that's not the point. The point of this class isn't to have you memorize all of these vocab words. Remember the point? At the very beginning, we said to make you a little more science literate in order for you to understand the why, or to consider the why behind some things. So why? What was important about this? Well, if we can remake it, if we can make this glucose molecule and regenerate RUBP and deal with that, this glucose molecule, this G3P, glyceraldehyde 3-phosphate, is a skeleton that we can use to make fatty acids. Glycerol that plant oils are from, simple sugars, complex sugars, fructose, like starch, cellulose, amino acids. Without photosynthesizers, we can't make all that we have to make. We can't make our own amino acids. We can't make our own base carbon skeletons. We have to consume things that make this from sunlight. Mammals don't have that ability. And so this picture helps us get a good view of how G3P is used. What are some ways that we can use it? And so G3P, glyceraldehyde 3-phosphate, can be used to make all these other things, all these other substances that we can use, and then put into the mitochondria to get energy if we have to. And so it's an end product that can be converted into food, energy, plant growth, cellulose, proteins, flowers, seeds, reproductive features. And so it's not just C6H12O6, it's this G3P, glyceraldehyde 3-phosphate, that we can use to form everything else in the plant. We have a way of storing high energy in the form of glycogen in our liver and muscles, and adipose tissue, lipid storage. Plants have a different system. Some plants store their excess glucose, excess energy in their roots, and we use that for food ourselves. And so some plants that have very large modified roots, like carrots, turnips, radishes, we consume those for that energy. Some plants have modified stems, like things we call tubers, like our potatoes. Modified stems just full of starch, full of energy that we can utilize. Some plants have modified leaves. They've turned their leaves into a way to store excess food, like your onions, your daffodils, your things with bulbs, like your lilies. And so we utilize that for food source, roots, stems, and leaves. Carrot's an example of a root, potato example of a stem, onion example of a leaf, if you're considering human food. And so what I've done here at the end, this is not the only type of photosynthesis. And in fact, we skip a lot of the importance of photosynthesis, but if you take botany, you'll get that in the future. And so what I've done is I've given a review. I broke it up into bullet points here at the end. The light-dependent reaction, the main outcomes. This can help you with that study worksheet. I break up the Calvin cycle into main bullet points. That can help you with the study worksheet. Because next week, we're going to look at other types of photosynthesis and then move into cellular respiration. So if this was a lot, and it is, I need you to use that study worksheet and use these two slides here to go through. Learn the basics first, know what the equation is, know what the outcome is, and then go and try to learn all the individual details. Listen to this again if you have to. Read that again if you have to. Use those YouTube videos that are posted. Use that study worksheet. Use the study guide. This is not a time to wait until the very last day of this unit. It's not going to cut it. I don't know if we'll have school tomorrow. Who knows? I can't see the future, right? But I'll be available through email. you know, give me some time to answer. I'm not always immediate. I don't keep my phone with me all the time, but email me if you have questions. People 4 years from now will not need to know this information, but I do have some exams graded. I'll put those in. They weren't the best. And so let's use this as a motivator to do better on this. Let me know if you have any questions, and then try to stay as warm as possible.